This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 97, recorded on January 22nd, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hi there. How you doing?
I'm well. We haven't been together on a TWiM for a few weeks.
That's true.
Maybe a few months. A month.
Uh-huh.
Maybe this is the first one this year. Yeah, it must be, because this is only the third week of January.
I guess.
Happy New Year.
Thank you.
Happy
Same to
New
you.
Year.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Happy New Year to you as well.
Yeah, nice to be back.
Yep, and everything is great in Ann Arbor.
Yep, it's pretty good. You know, we've got a nice coat of snow on the ground, but the sun has been out today. So,
how many inches of snow do you have?
Oh, maybe 2.5, 3.
Yeah, we have none here, but it has been deadly cold for the past few weeks. We actually went down to minus 15 Celsius.
Ouch.
Very cold. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. Happy New Year.
You too. Everything going well with you?
Everything's great here in beautiful South Carolina, where it's a balmy 61 degrees.
I don't want to hear it.
Out.
I don't want to hear it.
Yeah, we have what our Chamber of Commerce calls good weather.
It's not Chamber of Commerce weather. Huh?
No, it's just good weather. It needs to be about 10 degrees warmer, and then the golfers come out of the woodwork.
Elio, do you have rain out there, or was that it a couple of weeks ago?
Oh, that's a couple of weeks ago.
Now it's dry again, huh?
We need it again. There's some promise. Maybe El Nino will show up in these parts. But otherwise, I don't want to talk about the weather here, because if I do, everybody's going to want to come to San Diego and live here.
They already do.
And consume your water.
Yeah, they already do. It's very crowded there. Elio, you have your video on. If you want to turn it off, that would conserve bandwidth.
Okay.
I don't mind looking at you, but there you go. Thank you very much.
Sure.
Well, this is the first time we're all together for a long time. And we do have some cool things to talk about today. So let's get started. And first we'll hear from Elio with another snippet.
And a snippet means a short piece, right?
Yeah, now what's short? Is that like 5 minutes, 10 minutes? I guess
I have no idea.
Depends when he's done.
When I'm done, I'm done.
That's right.
All right. So the paper is entitled Gill Bacteria Enable a Novel Digestive Strategy in a Wood-Feeding Mollusk. And it has 22 authors, so I won't read them all. The last two are Margot Haygood, who is at the Oregon Health and Science University in Portland, and Daniel Distel, who's at the place that sounds intriguing, the Ocean Genome Legacy Center of New England Biolabs. I looked it up, and they do serious work there. Anyhow.
One of them is from your old place, right? Tufts?
Yeah. No, well, somebody's from Tufts, yeah. I'm not from... I don't know this person, Roberta O'Connor.
Yeah.
I haven't had the pleasure. Well, I've been gone a long time, so that's why. Anyhow, this is an interesting paper. It appeared in PNAS, and it deserved that kind of attention because it tells you of something new in the world of intestinal microbiology. I'll give you the punchline. Shipworms are the guys who eat wood, and they used to eat wooden ships. I'll come back to that in a minute. Okay?
Makes sense.
Right. But they do it, of course, with bacteria, because no animal can digest wood directly, cellulose and the other intricate polysaccharides of wood. But they do not have their bacteria in the cecum, like we do, and termites do, and beavers do, who are probably the only vertebrate that eats wood. And they don't do it that way. They put them instead, of all places, in their gills. And by gill, I mean gills like fish gills. It's a place where they exchange oxygen with seawater. Strange place. But the wood ends up in the cecum. So what gives here? Well, what gives is that the enzymes involved in wood degradation are made by the bacteria in the gills, and the enzymes are transferred to the cecum. Now, you've never heard of a thing like this. This is totally, it blew my mind because this is totally unheard of, unique, and why would they do that? So anyhow, let's go over it a little bit in detail. So first of all, shipworms. Shipworms are not worms, they're mollusks. They're like a snail, only an elongated snail. And boy, they get to be big. They can be as long as, you won't believe this, a meter long, although most of them are only a few centimeters. And in the old days, you certainly lost your whole armada to shipworms, unless in the, I think, 18th century, you learned to copper-clad the bottom of the wooden ships. And if you did that, you saved them. But otherwise, these guys would eat you out of house and home. And you'd have a leaky ship that pretty soon would sink. But they don't just do evil things like that, evil to the owners of wooden ships. They also help remove debris, wooden debris, say in a harbor where it's not wanted. They help recycle material, which is certainly important. So they have a lot of interesting activities which are not necessarily all bad. These guys use wood as their main food source. In fact, one of them has been studied that can live on wood alone. I thought we cannot live on bread alone. They live on wood alone.
I was trying to think of that.
Anyhow, there are a whole lot of them. There's a whole lot of different kinds of shipworms. And these people studied one particular kind, but they all have something, this idea in common, that if you look at the cecum, which is a good part of the body of the animal, you find that there are no bacteria, but very few bacteria. By microscopy, using fluorescent dyes, it's less than 1% of what you find in the gills. Well, no, in the gills it's not fair, but less than 1% of what you find elsewhere in the digestive system. So something actually cleans up the cecum of the bacteria that are normally you'd expect to get there because these guys don't eat sterile food. So then nobody knows what that is, whether there are special antibiotics or the equivalent of an acid stomach someplace, although I doubt that that's the case. In fact, it's not the case. But so they must disinfect the food that ends up in the cecum by some mechanism, and what's in the cecum would sit there forever undigested, were it not for the bacteria in the gills. Okay? So this is actually... let's say, pretty startling. And in the gills, the bacteria are found in bacteriocytes, just like the cells, the specialized cells of insects that carry bacteria. And within the bacteriocytes of the gills, the bacteria are packed as densely as you can. They're wall-to-wall bacteria. Okay, so far?
Mm-hmm.
So the bacteria in the gills, the enzymes are in the cecum, and the wood is in the cecum.
So you said there are no bacteria in the cecum?
Almost no bacteria.
Almost no? Okay.
Yeah, very few. I mean, it's nothing like what you find in termites or in people, which is
remarkable in and of itself, isn't it?
Yeah, as I say, I don't know how it happened. There must be some way of disinfecting the food before it gets to the cecum. And there's no mention of how this may happen. At least I haven't run into it. Okay, so what bacteria do you find in the gills? So you find it's a relatively small number of them, and they are anaerobes with strange names like, let me see, a bug called Pteridinobacter. Teredo is the name for a shipworm. So Pteridinobacter is a bacterium that carries that is involved in this. It's, as I say, it's found in the gills in large numbers, and there's a few others that make up about most of the bacteria in the gills are just a few kinds. 4 strains make up most of them, most of the microbiome of the gills or the shipworm. And if you look at their genes, you find that they are loaded with enzymes that are putative decomposers of carbohydrates, of polysaccharides, glycoside, hydrolases, carbohydrate esterases, polysaccharide lyases, and so forth. So they have maybe a couple of hundred genes for these enzymes. And if you look at the proteome of the gills, you find something, and of the cecum, you find something quite interesting. In the cecum, in the gills, there are 102 proteins that are identified, and they represent everything, all aspects of metabolism of a bacterium, biosynthesis, energy, and so forth. Everything you can think of is represented, and these polysaccharide degrading, putative polysaccharide degrading genes make up only about 11% of all the proteins. The proteins make up 11% of the proteome. Now you go to the cecum and you find 42 proteins, and 41 of them are involved in polysaccharide degradation. Okay? So there is it's almost like a pure solution of carbohydrate degrading enzymes in the cecum.
So how does the enzyme that degrades the lignin associated with the wood, I thought that was principally associated with the fungal group, principally Phenerichidae. So do these bacteria have ligninases?
I listen, I say it's a very good question. I wondered about it. I didn't see a mention of lignin in this paper. So it's as if lignin does not get degraded, but gets excreted. So the story of the fungi are not, best I know, are not here. And the degradation is mainly of other polysaccharides. Well, lignin is not a polysaccharide. Is of polysaccharides. In other words, cellulose, which is the main one, and all the others, pectins and so forth, which make up the wood. So we're talking here about degradation of only part of the wood, I guess, because, as I say, I didn't see lignin mentioned. Maybe it's there, but I didn't.
And in a boat, it doesn't really matter if you destroy the lignin because you'll still have water in the boat from eating the polysaccharide.
Right, exactly. They think it's squishy and
you sink.
It's a kind of rot. Anyhow. But the curious thing is that the cecum has only 42 proteins, and 41 of them are involved in this thing. So it looks like a pure solution. Okay? Now, how do they get there? Well, that's a good question. They don't know. The gills and the cecum are nearby in this animal. They're almost touching end to end, but that doesn't help. There is a structure called the Des duct, which goes from the gills to the esophagus, which is not the cecum, but it's on the way to the cecum. So maybe that's a duct whose function is not known. Maybe the bacterial enzymes are made in the gills and travel via something to the cecum. Now, the bacteria certainly secrete these proteins. They certainly have, the enzymes have secretion signal sequences, so they can get into the periplasm of the bacteria. They escape from the bacteria, they escape from the bacteriocyte, and somehow they are deposited in rather large amounts in the cecum. Now, that is weird. So it's like they like their enzymes neat.
It really gives a whole new perspective to trust. This is truly trust on an evolutionary level. I mean, you're trusting that protein will be put to good use. So what's the selective advantage to the microbe to export this protein, aside from living on the gill, where it can probably dump all its waste electrons and get respired away? But from the animal's perspective, how that coevolution of secreting large volumes of proteins in order to sufficiently turn over the polysaccharides associated with wood is just got to be a tremendous selective pressure.
Sure. Well, let me propose something. They kind of mentioned this. It is possible. First of all, this is rare in nature, but it has happened that in an aphid, an insect, a pea aphid, proteins from the endosymbiont find their way into the saliva and the honeydew of the insects. That's the only other case where enzymes are exported in purified form, essentially. But here, the case you can make is that this may be more efficient. That is, in a cecum where you have nothing but enzymes, powerful enzymes that destroy what you're trying to destroy, you're better off than having a whole microbiota full of who knows what all, including proteases that may destroy the enzymes you need. So this is sequestering the enzymes you want in purified form where they're not going to be bothered by anybody. Now, I'm just mouthing this. This is just words. But you can make that case. And so this would have a selective advantage because it would be more efficient from the point of view of the shipworm. It would be a more efficient way of getting the food, of breaking down the wood. But as I say, we don't know. We're left with that.
It's puzzling.
It is
applications.
Puzzling. Sorry?
I was interested in the closing paragraph of their paper where they talked about applications of this knowledge.
Yeah, well, they say that these enzymes must be pretty good. And so since people are looking for ways of breaking down cellulose so they can make something, useful from that, these may be enzymes that should be looked at. And I imagine people are doing that. You can certainly clone the genes for these enzymes into something, and you can probably produce them in large amounts. So this may be better enzymes. Yeah, it's a good idea. I mean, I like the thought. So decomposing plant cell wall biopolymers and wood is a way to make eventually what's called cellulosic biofuel, among other things. So... And these enzymes must be really good at doing it. There's no question.
And stable, and probably stable, because you have to anticipate that if you're sending an enzyme down a pipe, and you are anticipating that it's going to do real work, its turnover rate is probably much slower than an enzyme that's living in your garden variety bacterium cytosol, where turnover could be much higher, especially in these oxidative reduction reactions where part of the redox balance is generating free radicals, which often degrade proteins in situ. And so that may be one of the selective pressures for the microbe, is to give the enzyme away, allowing the symbiote to effectively send back carbon and energy to the microbe from a much more stable situation, uncoupling the oxidative risk of metabolism.
That's right. It may be that way. But somehow I find this puzzling and interesting, and it may end up being something useful. There's another point I make in the last paragraph, namely that having just relatively few proteins, makes it for a better industrial cocktail, an industrial gemisch of enzymes, because this cocktail that's in the cecum of the shipworm works.
Right.
And whereas if you look at termites, you're going to find such an immense number of enzymes, it's going to be hard to sort out which ones work with which.
So the microbes will show the way.
Yes, the microbes will show the way.
So there are other examples where bacterial products are transported elsewhere, but is this the only one where it's to a very specific place that we know of?
Well, as I say, except for the aphids that I mentioned, which end up where some of bacteria, the endosymbiont of the aphids make proteins that end up in the saliva.
Yeah.
And that's the only other example that they quote. So I don't know of any other.
We'll get letters of people telling us about exotoxins that bacteria throw out and end up in our bloodstream.
For sure, they make plenty of bacteria, secrete plenty of proteins, but this particular combination, I think, is quite unique.
But they don't help us much, these exotoxins, right, Michael?
No.
Yeah, I'm thinking of beneficial things. Anyway, that's very cool, and I think the title is just neat with all these words that you don't usually see in the title. So, Michael,
I think they should have said, "I like my enzymes neat."
I like my enzymes neat.
That's actually a good title. I'm going to write that there. Michael, that copper on the bottom was great to keep the bacteria away. Huh?
Absolutely, and in fact, it's not only bacteria that copper inhibits; it inhibits everything else because it's toxic for the reason that it's going to serve as an electron sink, and it's going to uncouple the same membrane function that bacteria require and anticipate. It's going to happen in eukaryotes because there is the universality of the biochemistry.
Yep. All right. Thank you, Elio. Cool snippet. I like that.
Thank you.
We have a paper now which Michael will tell us about, and Michael had sent this out for our checking out a week or so ago, but it turns out that we had an email from Dennis who sent a link to this paper, and he said, "Is it too early for a TWiM on this?" And I guess it's not, because we all decided to do it. So, Michael, take it away.
Well, the title of the paper is "A New Antibiotic Kills Pathogens Without Detectable Resistance." So, first off, you get that it's a provocative title. Without detectable resistance is sort of heresy when you talk to your infectious disease friends, because they bring up the Star Trek quote, "resistance is futile," because bacteria will become resistant to almost everything. And so the paper is by a group of authors from a number of different places in and around Boston. And the first author is Lucy L. Ling, followed by Tanya Snyder, Aaron Peoples, Amy Sporing, Ina Ingalls, Brian Conlon, Anna Mueller, Till Chaberlet, Dallas Hughes, Slava Epstein, Michael Jones, Linos Lazardes, Victoria Stedman, Douglas Cohen, Cynthia Felix, K. Ashley Fetterman, William P. Millett, Anthony Nitti, Ashley Zullo, Chao Chen, and Kim Lewis.
And I'm sorry, we're out of time.
Well, the reason I read all the authors is I think this is a really clever paper. So I'm going to set the stage for you. So
I need to add that some of them are from Germany, and then some are from biotech.
Yeah.
Right.
They're in the Boston area. So this is a really neat public-private international collaboration.
And they start out their abstract by setting the stage for the reader, and this appeared in Nature a few weeks ago. And the first thing they tell us is that antibiotic resistance is spreading faster than the introduction of new compounds into clinical practice, causing a public health crisis. And one only needs to Google search the Longitude Prize that's coming out of the United Kingdom, where they're going to give someone £10 million to help solve the problem of global antibiotic resistance. And then another project that's being funded out of the Infectious Disease Society, which is called 10 by 20. And so the professional organizations are very much aware of the crisis that we're facing with finding new antibiotics. And the argument that the authors eloquently make in one sentence is most antibiotics were produced by screening soil microbes, but this limited resource of cultivable bacteria was overmined by the 1960s. And to some extent, that's true. And then they go on further to state, arguing that the synthetic approaches to produce antibiotics by high-throughput screening and some of the strategies that have been successful in developing ethical pharmaceuticals for human use to specifically target our products, like reactive G proteins and whatnot, have been unsuccessful. And then the fourth piece, which takes us into the clever aspect of their paper, is they estimate that the uncultured microbes make up approximately 99% of all the species in external environments. And these untapped or uncultured microbes are thought to be a source of new antibiotics. And I really think this is the crux of the hunt. And if you think about it, microbes have been aggressively competing for the same resources for at least 3 to 4.5 billion years. And how long have microbes been around?
About 3 billion, but not 4. That's about it. I mean, the Earth started about 4.5 billion years ago.
Yeah, but there, if you
it didn't start right away. There was a billion years where there was no.
There was nothing. So in this paper, the authors developed several methods to grow uncultured organisms, and the technique they used was first published and applied in environmental microbiology, and it was called the iChip. And the iChip was developed in 2009 when everyone was going crazy with the apps that were being developed for the iPhone platform, and everybody was walking around saying there's an app for that, and everything had the prefix I. So it was called the iChip. And using this method, and I'll describe that in the meat of the paper, the authors described the discovery and the characterization of this new antibiotic that they called tioxobactin, that they selected from a screen of uncultured bacteria. Up until that point in time, most of our antibiotics we got from dirt, we isolated in pure culture, we grew them, and then we began to ask the question in almost the same manner that the authors do of, is there a zone of inhibition around a distaffusion assay? And so to give you the final take-home messages, this new antibiotic inhibits one of the primary targets for a lot of antibiotics out there, namely cell wall biosynthesis. And what tioxobactin does is it binds to a highly conserved motif of 2 precursors that are involved in cell wall biosynthesis in bacteria. And the cell wall is such a tremendous target for antibiotics because it is very limited in its ability to cause side effects in human hosts, simply because we don't have any peptidoglycan in any of our cells. We don't have cell walls. And so if you're going to make a drug that you want minimal side effects against or from, you will effectively target a structure that is absent in your in the individual that's going to be taking the drugs. And so the last piece that they nail us in their abstract is they report that they failed to they did not obtain any mutants of Staph aureus or Mycobacterium tuberculosis that were resistant to tioxobactin. And the significance of this is the properties, and they give us wonderful looking chemical structures, and the properties of this compound suggest the path forward for other workers in the field to develop antibiotics that are likely to avoid the development of resistance. So here's the story. Question 1. How did they select for this drug? And their working hypothesis is that there's gold in them dare hills. They're saying that we have all these opportunities. And this is what I always get picked on by my classmates from graduate school, because once I was teaching a class and I asked the other teaching assistant, where do we get dirt? And we were basically hunting for, unique bacteria. And so the other teaching assistant, Cheryl Blake, said to me, you just go outside. There's plenty of dirt out there. And so I get picked on to this day about where do you get dirt. But this is the real crux, and this is the story of the iChip that the worker on this paper, Epstein, actually developed at Northeastern University in Boston. And the way I look at this is microbes are smarter than us. They don't order from the DIFCO catalog or Sigma. They get their building blocks from their environments. They are, if you will, the original off-the-grid survivalist. And so what Epstein and his colleagues did at Northeastern was they coined this term, the iChip, for high throughput in situ cultivation of these unculturable species. And quite frankly,
uncultivated, not unculturable. They make a point of that in one of their papers.
Yes.
These guys grow someplace, so it's our lack of ability to cultivate is our problem.
Heretofore uncultivated.
Okay. So microbes are hard to grow. And so when I was reading this for the first time, I remembered the first lab project I was offered as a graduate student, and that was to develop a working medium to cultivate magnetotactic cocci that were growing in freshwater sediment that my mentor, Walt Konietzko, literally pulled out of Indiana ponds. And he was trying to enforce on me how challenging it was to begin to work out a culture medium to grow a microbe that literally swam out of the sediment. And when I was in graduate school, he recruited his old undergraduate student, who was Norm Pace, to help him work things out. Norm Pace is sort of my long-lost brother, if you will. Norm was did his undergraduate research in Walt's lab at Indiana, and so Walt recruited him back to Indiana to join the faculty. And so this was at the time that Pace was working with Woese on developing the technology from 5S to 16S. And since the way Walt isolated the magnetotactic bacteria is he would put a magnetic stirring bar next to the glass, and the magnetotactic bacteria would literally be attracted to the pole of the magnet, and he'd sneak past her pipette down there and suck out the bacteria. And then what Norm would do is he would isolate the 5S RNA or 16S RNA, because this was before PCR, and he would then sequence it. So Walt could figure out at least where these microbes were so that he could figure out how to develop a medium. Now here, the authors with their iChip use similar methodologies, understanding their community, and what they do is they take suspension, and let's just say it's dirt, and they suspend it so that the concentration of bacteria that they will place between the screen will be the equivalent of one bacterium per the hole in the screen. And then they load in agar to effectively immobilize the microbe, if you will. If you imagine this looking like a microtiter plate, and you have one microbe in one well that you have an agar plug on top of, and appreciating that the agar has no nutritive value to the majority of microbes on the planet, you then simply dip this slide in a slurry of stuff, whether it be the dirt that you got it from, or you could concoct a medium. And what these authors did is they just literally concocted a medium, and they were able to isolate this particular microbe that they are basing this paper on, and they were able to isolate gram-negative microorganism that is capable of producing this antibiotic. Now, if you think about it, when you read the old micro textbooks, this technique of the iChip is really nothing more than the old agar shake tube, where you would take a soil sample, you would then dilute it to extinction, and you would literally see microcolonies growing in these cylinders of agar, and then you would take a razor blade and you would literally cut out the colony from the razor blade of where these things are. And we've all seen the agar shake tubes because it's effectively part of the introductory microbiology lecture.
But the elegance of this technique is they say, I'm not going to spend X number of months or years trying to figure out an optimal media to culture these. I'm just going to stick it right back in the soil, let them grow, and then once I have numbers, I can do my biochemistry and my microbiology.
And
so
that's
I thought that was
fabulous.
Mm-hmm.
It's the head slap experiment.
Right.
Why didn't I think of that? You literally want to slap yourself in the head. And so the authors estimate that this method will allow them to cultivate at least 50% as opposed to the 1% of microbes we presently can cultivate using garden variety media that we may have in our lab.
Michael, this is the way to cultivate all those gut microbes. Just
absolutely
put a plate in someone's intestine. Wow.
You literally have that. I think you're going to be able to do this, and in fact, the gastroenterologists are already doing this to some extent, Vincent, when they give you these camera probes that you swallow and they swim through. Mm-hmm. Just imagine attaching a screen to this to pull the sample and leaving the camera down there for a while and allowing it to move out. And as Michele said, they then did their biochemistry. They were able to cultivate them in vitro. And now,
by the way, one interesting thing is that when you go through this procedure, the bugs that grow in the eye chip, many of them are now capable of growing on agar plates and give you colonies. Right. So they have to sort of be coached into learning how to grow on an agar plate by going through the eye chip procedure.
Yeah, I guess it's an adaptation effect. And this is not news to the pathogenic microbiologists, because they know that when you take a microbe out of an animal that's infected, it behaves very differently, or the biochemistry is very different than when you isolate that organism off of a Petri plate. It effectively has lost some of its fangs as it passes serially on agar plates. It's probably just simply turning off genes. So jumping ahead, what they were able to do is they were able to cultivate these organisms, and then they isolated, supernatants and fractionated and did all sorts of other remarkable things, and they put them onto a lawn of Staph aureus.
Wait a minute, they did it a little bit simpler. They put a lawn of Staph aureus on top of the eye chips.
Right.
And they saw which ones directly.
And they did it directly, asking the question. I was trying to go how they pulled this antibiotic out, but Elio's absolutely right. They did it really elegantly. They just slapped the eye chip on top of a lawn of Staph aureus and asked, is there a zone of inhibition? And you literally take it out of the liquid and you literally put it on top of a lawn, and if the little well was making the antimicrobial that was diffusible, it would diffuse into the agar and inhibit the growth of Staph aureus, which was their intended target, because they were looking for an antibiotic that could inhibit the scourge of most ICUs, which is MRSA.
And let me just point out, by having such a simple, small-scale test, it allows them to quickly and relatively cheaply screen hundreds and hundreds of different microbes.
Or
so
soil
I think
samples.
That's the elegance, to have that quick screening overlay assay.
And it goes back to what I get picked on. Where do you get dirt? Because now you can have your friends go and get dirt from everywhere. And in 1990, I learned the statistic that there were about 10,000 species in a gram of fertile soil. And then Schloss and our good friend Joe Handelsman in 2006 postulated that there were between 2,000 and 8.3 million species for soil microbial diversity. So now you can begin to appreciate the power of this technique for hunting for new antibiotics.
Michael, can I ask you, when they I'm tracking back a bit, but when they put this in the soil, where does this in the laboratory? They've brought soil into the lab, or do they go put it outside? It's not clear. It's not clear to me, because
it wasn't clear to me in the paper. But I guess you could do it both ways, Vincent.
If you have a protected area outside, we don't know where no one's going to walk on it or steal the plates,
Or you bury it. You bury it in the dirt.
Yeah, you could bury it, I suppose.
I think that's what they did, actually.
And the other advantage, again, because we all breathe air, we like to work with aerobes. And unless you happen to have an anaerobic chamber in your laboratory, it's very challenging to work with anaerobes, whereas soil will quickly become anaerobic because of the intense competition of all the different microbes pulling the oxygen out. And so you can actually set the proper redox potential, and which is the hardest thing to do for developing bacteriological media, setting the redox potential. It's orders of magnitude more complicated than just pHing your media.
So they got this. They got the strains or the bacteria from a soil sample in Maine, right?
Correct.
I wonder if it's important to incubate these plates in the same soil where you got that, right? Or could they do it in their backyard in Boston? Because
they could probably do it in their backyard in Boston if it's the right loam. But if you've ever talked to a soil scientist, you'll get the lecture that not all dirt is dirt.
Yeah, I can understand that. Yeah,
and we'll get letters from the soil scientists if we say dirt is dirt. And so, but the other aspect of this is because of, and this is the trick of the iChip technique, is they dilute to extinction where there's one cell in these wells. And so that's the trick is, again, it's the base of all pure culture technology. And you can then isolate, once the cell has grown up to numbers, you can isolate, and then using the universe primers for 16S, you can effectively either do whole genome sequencing using a shotgun approach, or you can just figure it out using the 16S ribosomal database, what you have. And that's effectively what they did. They sequenced the parent and learned based on 16S ribosomal RNA and then in silico. And in silico DNA hybridization. Doesn't anyone work with DNA anymore? It's all done with the computer, I guess.
We're out of jobs, yep.
This organism belongs to a new genus related to aqua bacteria, a genus not known previously to produce an antibiotic. And Aquabacter is a gram-negative microbe, which will become important. So now they can do all the fancy high-throughput mass spectroscopy to figure out what the magic elixir was associated with the iChip that said, yes, let's screen this. And I'm not going to go through all of the fancy biochemistry, but they determined that tioxobactin was a 1242 Dalton depsipeptide, which contains enduracidine, methylphenylalanine, and 4 D-amino acids. Now, I had never heard of endoduracidine, but I've heard of D-amino acids. And every first-year microbiology students know the only place you find D-amino acids in living tissue or living cells is in bacterial cell walls. So I immediately guessed the target, even before I got into the meat of the paper, was the cell wall, because of the fact that in Staph aureus, their original target organism, it's got a pentaglycine is what forms the cross-linking. So this forces us all to remember the god-awful peptidoglycan synthesis, which is a complicated process done while the cell is growing. And the way I explain this to my dental students is I point out to them, some of them are in the Navy, so I point out to them, consider a submarine making itself bigger while moving. How do you go about doing that? And that's effectively what the bacterium does. The bacterium has pressure in the boat, just like a sub. Bacteria are at about 80 psi. And then using a disaccharide of N-acetylglucosamine and N-acetylmuramic acid and a beta-1,4 linkage, they extend this as a polymer to make the structure that we know as peptidoglycan or murine. And the way I explain this to my dental students and my medical students is you need 3 components to make cell wall, and their cell wall is assembled in 5 steps. The first 3 occur in the cytoplasm. Step 1, you synthesize the disaccharide, which is the muramic acid and the N-acetylglucosamine. And that's coupled to UDP, uracil diphosphate. Then what you tack onto that muramic acid and acetylglucosamine dipeptide, you effectively, disaccharide, you tack onto it the cross-linking members, because remember that peptidoglycan lends its rigidity to the cell by effectively cross-linking it. And most entering students to microbiology don't really understand what cross-linking is, but most folks have seen a nylon stocking, and they know that the linkage between the nylon stocking enables it to have strength. And this strength comes from the peptide bond that forms between the two strands, and it's that cross-linking peptide that gives the rigidity to the cell wall. So this 5-step process of this cross-linking is what's critical to understanding the intent of the second clause in their title, namely the concept of without detectable resistance. And so what happens here is it's the association with the lipid carrier. And to make the long story short, this is where tectinobactin interferes. It interferes with the lipid precursor as a stoichiometric competitor to the lipid precursor. And so consequently, as the sugar is moved from the inside to the outside, this drug will interfere with the movement of these precursors, and the net consequence is you can't make additional cell wall. And so these two lipid precursors, the fact that this antibiotic, tectinobactin, binds to lipid precursor 2 of peptidoglycan and lipid 3, both of which are associated with gram-positives. So the fact that this drug binds multiple targets is likely how it takes us to the author's claim of the mechanism of exclusion. And when you look, you see that this drug is very active and effective against gram-positives. It's effective in concentrations, for example, methicillin-resistant Staph aureus. It's effective at 250 nanomoles per milliliter, while when you look at the gram-negatives, it requires a much larger concentration in order to inhibit the growth. And that is because the gram-negatives have an outer membrane, and because of the structure of tectinobactin, it's unable to get into the cell. The way the gram-negative aquabacterium makes this drug is it exports it out of the cell completely. And consequently, gram-negatives cannot bring it into the cell. So this is a selective antibiotic that appears to be selective against gram-positives. The other very important issue is that this antibiotic is bactericidal. And remember how you tell if something is bactericidal or static is you look for death of the population. You monitor the viability and not the number. So you have to do a viable count as opposed to looking at it in a spectrophotometer. And that's effectively what these authors have done. Is the first question they ask, is it bactericidal? And they did this by harvesting early log phase growth cells. And they ask, is it bacteriostatic, which will give them a flat line in terms of population, or is there killing? And they learned that this drug, like vancomycin and oxacillin, was cidal. But here's the magic of this antibiotic. They actually then took late log phase bacteria and asked the question, would this drug work against this community? And the answer was yes. Normally, drugs like penicillin and vancomycin are most effective against aggressively and quickly growing bacteria. And that is why penicillin is not very effective against TB, because tuberculosis is really a very slow grower. But teixinobactin works against the community, and in late log, the majority of cells have stopped growing. And the fact that it is still bactericidal is good news for the physicians who are listening, because it means that for skin and soft tissue infections or activity against tuberculosis or any slow-growing infections, it will have activity.
And Michael, I was able to correspond with one of the team members, Brian Conlon, who's a postdoc in Kim Lewis's lab, and he said pointed out exactly what you mentioned, that the day they saw the bacteria lysing was an especially exciting day in the lab, because they realized they had their hands on something special. So Brian is going to continue to work on the mechanism of how that killing works as he launches his own independent career.
And it's
nice.
that lysis that is so important, because it now enables you to think about how to develop a high-throughput screen, because you can use optical density rather than viable counts, which are much more labor-intensive and expensive to do. And then the other question that these authors asked, and this is what was done in the 60s, looking to answer the question, how quickly will resistance emerge, is you grow the population in a sub-lethal or sub-MIC concentration, and then quickly ask how fast the bacterial population will escape the effect of the drug. And what they learned here is that they did not see escape of the population incubated in teixinobactin, but they did see escape when they grew things in oxafloxacin, which is another peptidoglycan inhibitor. And this was really exciting news to me as well. And I think it goes back to the peptidoglycan story, is that this drug has two targets. And so when you think about it, this beautiful drug is, inhibiting this. And then they ask other questions. Does it inhibit other cell processes? And the answer was effectively no. And they're effectively asking toxicity questions and general utility questions. That's not really germane to the story, because I think everyone out there wants to know, will it cure an infection? And that's
what they did. Yeah, you're going to talk about the animal experiments now.
I'm going to talk about the animal experiments now. So in the animal experiments, they're asking the question simply, will this work in mammals? And they used three different animal models, and the control drug that they compared it to was vancomycin. And you typically do these drugs by giving them the concentration drug measured against the weight of the animal. And what they learned is that in single-dose treatment, IV, 1 hour post-infection with 6 mice per group, and this is pretty much how one does these sorts of experiments, that produce septicemia, or you have a fulminant infection in the blood using methicillin-resistant Staph aureus, or MRSA. They then simply ask the question, is the mouse alive at 48 hours after infections? And what they learned is remarkably, at a very low concentration, between 250 micrograms per milligram per kilogram of animal to 500 micrograms per kilogram of animal, they saw significance in that there was no deaths, 100% survival. They used two other models to effectively ask the question. One, skin and soft tissue model, which is where they, again, giving a single dose, 2 hours post-infection, where the mouse, this was a neutropenic mouse, and a neutropenic mouse is important because the neutrophils will effectively eat the bacteria and clear it out. And so if you're neutropenic, you're deficient in neutrophils, the infection is just going to take much more quickly. And again, this new drug, teixinobactin, was effective in protecting the animals. And in the third experiment, again, this was a 2-dose treatment. They again saw in immunocompetent lung model infection using strep pneumoniae, and they were measuring lung colony forming units of bacteria of strep pneumo determined after 48 hours post-infection. And the colony forming units for each mouse show that you effectively have literally 1 billion strep pneumo in the lung in the animal with no drugs after 2 hours. And then when you give this drug, you see that the concentration of bacteria are effectively down from 10 to the 9 bacteria. The majority of them are between 1,000 and 10,000 bacteria per animal, or excuse me, per lung. So I look at this and I say, my goodness, this really has activity in that the drug is able to move IV in the animal and deliver to a target and actually cure an infection that is normally lethal to this animal. So I think they have something here. The remainder of the paper goes into the most provocative aspect, is whether or not bacteria will develop resistance to this new drug. And what the authors point out is it took over 40 years for vancomycin resistance to come about. And vanc acts very similarly to this drug, but not on the same target. Vancomycin competitively will interact with the peptide moiety of the peptidoglycan, which is why I was going through the synthetic aspects. But the fact that there's 2 targets for this antibiotic is going to complicate the emergence of resistance, because what the cell needs to do is to reinvent cell wall biosynthesis.
Although, to their credit, they point out a different mechanism of resistance, which would be an antibiotic that would cleave this molecule.
Right.
Rather than changing the target.
Yeah.
Yeah, and also the vancomycin argument, I think, is not foolproof. Vancomycin is a fairly toxic drug, and it has not been used over the last 40 years to the extent that other ones have. So it's not quite fair to say that it's intrinsic to it, that resistance took 40 years to develop. Part of it is it wasn't used much.
And it was originally extremely challenging to synthesize. In its first incarnation, vancomycin was referred to as Mississippi mud, simply because it literally looked like they were injecting mud into the patient's vein. And
so it may not be quite a fair analogy, but you hope. I think that I wouldn't put my money on bugs not developing resistance. And as Michele said, you can cleave the drug, you can prevent its entry. There are other mechanisms than the target.
Yeah.
And so I would not be so sanguine about that fact. But on the other hand, I think that the idea that most of what's out there potentially making antibiotics has not been looked at is right spot on. And I think the technology of the iChip is so powerful that they're going to find something. By the way, in another paper, they found that they can cultivate certain strains by growing them together with others, because in nature, probably one of the reasons you cannot cultivate bacteria singly is because they don't grow singly in nature. Yeah. They grow as a consortium. So if you favor the formation of consortia in the iChip, you're going to find many more things, and I'm sanguine about that. I think they're going to find a lot of antibiotics this way.
I think you're absolutely right, and I think they wanted to lay out the complete story, and I think they were intentionally provocative about the emergence of resistance. Back in the heyday of antibiotic development, growing candidate antibiotics in sublethal concentrations with the intended microbe was a standard practice, because drug companies were always wondering how quickly, or the shelf life, if you will, of their antibiotic in formulary, because if a population quickly becomes resistant to it, it's of limited value, because by the time you get the clinical trial done, the bugs are going to be resistant to it.
Exactly.
But the thing I was most excited about is the fact that the drug was active against tuberculosis, and the fact that it didn't require a substantially greater concentration than what we saw for Staph aureus. That's what was so encouraging about this candidate drug, is because as we did a TWiM already, all the drugs that are in the market for TB, in India at least, all the tuberculosis have effectively figured out the workaround for the drugs that we're currently using. So there's hope if this candidate drug may be useful. But, as Michele pointed out, resistance will likely emerge. It's just a matter of not if, but a matter of when.
So I just found in the methods that they say the iChip is placed in the soil from which the sample originated.
Smart.
Yeah.
So they must have put it in that grassy field in Maine. So Brian didn't mention that to you at all, did he?
No, we only corresponded just briefly.
Yeah.
But I have a colleague here at the University of Michigan, David Sherman, who's taking a similar approach, looking for natural compounds. David is at the University of Michigan. He trained at Minnesota, but he goes diving in the most exotic, wonderful places. So he is in warm waters looking for new bacteria. So I think when we expand our reach into these more exotic environments and use this technology like the iChip, there's great hope. And
actually,
this story,
right here at UC San Diego, in the Scripps Institute of Oceanography, there's really a large effort geared towards studying actinomycetes from the ocean, most of which had not been cultivated before. And since actinomycetes are such great chemists and they make so many of the useful antibiotics which have been useful, it's likely that that's a very rich source. And sure enough, they have found a series of compounds already, including anti-cancer drugs.
So that there's an accompanying commentary in Nature, and the title is An Irresistible Newcomer.
This was also picked up, this research article was picked up by the BBC, by the CBS, by Forbes, the Wall Street Journal. So it's generated a lot of excitement.
I just have to mention, my wife used to work at Merck, and years ago she was involved in an antimicrobial program. And whenever we would travel, she would bring a sample collection kit with her, which consisted of Ziploc bags, a little shovel, and this was before cell phones. It was a little thing that you could get to read your longitude and latitude, because they wanted to know exactly where it was in case it worked, and they could go back to it. And they would bring it to the lab, but they would try and culture the microorganisms from this sample. It's very different from what was done here.
I want to go back to 1948, 49. As a kid, I lived in Ecuador. I used to work for a pharmaceutical company that had an affiliation with Imperial Chemical Industries in Britain. And one of my jobs was to take little jars, metal jars, and fill them up with dirt and send them back to London.
Nice.
For just this
There
purpose.
you go.
So it goes
See,
back
where do you get dirt's important.
Well, I tell you, Merck canned that was called the Natural Products Division, and a few years later they disbanded it because they were, as the article says, they weren't getting anywhere.
But didn't Merck just buy Cubis?
They just did $9 billion, I think. Yeah.
Yeah, so now Merck has an active antimicrobial program.
Yeah. Yep. Everything goes in
the
circles.
problem has gotten bigger. That's why.
Yeah.
Now we are under the threat of really drug resistance and bacteria being a very big challenge to society, and it didn't used to be. So now there is a reason for looking for drugs.
And with technological breakthroughs like this,
That's right.
and our ability to do deep sequencing, I mean, there's a lot of hope for microbiology to come up with some new natural compounds.
So I think this is a wonderful paper, but I would bet that this particular compound doesn't make it to market, but maybe a derivative or something
Right.
else.
Sure.
As Lynn
That's the story of drugs.
Lynn Enquist always used to tell me, Lynn is a virologist at Princeton, we all know, but he used to work at DuPont, and he said if you publish a compound, it means you're not going to pursue it and you're already onto something else. So,
and they publish the structure, and the structure gives us a great clue as to probably how the chemists are pulling their hair out, trying to figure out how this gram-negative was able to make this schizophrenic-looking molecule.
Yeah. All right. Thank you, Michael.
Thank you.
Let me just read a few quick emails here. The first is from Dennis, who writes, "Hi, Vincent and the TWiM-tastics. Thank you for your Virology 101 course. In Lecture 5 and the slide on estimated speeds of viruses through the cytoplasm, I was struck by the difference between the velocity of the bacteria Listeria monocytogenes with its actin tails in the cytoplasm versus these more sedate diffusion times. Given gene sharing, I wonder why more bacteria haven't been discovered using that mechanism. I learned of LM from Michele's discussion of Dr. Portnoy's Listeria work in TWiM 79. It is so fascinating and possibly world health changing. Perhaps Dr. Portnoy could be invited to do a TWiM." And then he links to two YouTube videos showing Listeria racing around a cell, and then another one, a presentation by Portnoy talking about his work. Michele might have already posted one or both of these. Thanks so much. You have set an example to which all podcasters should strive and a model that many would do well to adopt. 14 degrees C here and light rain, which we really need. Hoping the Northeast can dig out from its record snowfall. So I've been saving this for you, Michele.
Well, thank you.
But I don't remember having a record snowfall. Must be. Can this be last year? Can't be.
Well, the TWiM he's referring to was our review of the last general meeting, which would have been, last May?
Last May. Yeah, maybe it was as early. Couldn't it be? I don't understand. Anyway, so I talk about in my lecture that he's referring to how if viruses just had to diffuse through the cytoplasm, they would never get to where they needed to go. And so they employ motors to move around. But I guess Listeria moves very quickly, right?
It sure does.
The next one is from Dave, who writes, R.E., the letter which was under discussion when I came into TWiV just now in the last few minutes. Sensitive science fiction author Clifford D. Simak published a story some 50-some years ago about a peaceful society and how it happened that they could be that way. Every so many generations, they would go ask ceremonial questions of the omniscient computer after carrying on life as usual for a goodly time. First question was, what is the purpose of the universe? Answer, the universe has no purpose, it just is. Second question was, what is the meaning of life? Answer, life has no meaning, it is an accident on the face of the universe. After that, everybody could go home, go to the rocking chairs on the porch, and resume shucking the peas while telling each other entertaining stories about life, the universe, and everything. Certainly nothing to fight about given sustainable resources. Unseasonably hot October in Fresno, no rain. P.S. Now that the show's over today on Science 360, I'm going to have to go get the podcast for myself. I'm lazy and prefer to have a DJ bring it to me while I'm shucking the peas. Then he added later, he said, I think the two questions make more sense the other way around. I got it backwards. 1, meaning life is an accident. 2, purpose, the universe just is. Think so? I don't know.
The trip is what you make it.
It's right. I just don't know, Dave. I have no answers, but I suspect I think life is only here on Earth, but who knows? People tell me I'm wrong. Jim writes, in light of the discussion about C. elegans in TWiM 89, have any of you seen the open worm? I just ran across it. Yes. So this is a cool project where they're building a digital worm. So they've taken all the knowledge about C. elegans, all the networks of neurons and so forth, and they are trying to encode it in a computer. And over on TWiV, someone wrote to us where they have already used this information to build a robot, which acts a little bit like C. elegans. You give it something and it moves towards it or away from it and so forth. So that's pretty cool. Steve writes, hi, Vincent. As a mostly bed-bound sick person, I really must thank you and all your team at Twix for helping me to keep my sanity with your wonderfully absorbing and entertaining podcasts. With your frequent asides into seemingly almost any subject under the sun and out into the depths of the universe, distant past, present, and speculated future, you keep my mind as active as it could be, and it helps me feel a part of the real world away from the confines of my room. I'm even beginning to form a mental map of the U.S., its wildlife, and even weather, which is something I've always felt I needed to study more. I love the idea of the nitrochondrion, and we'll need a few more listens to take it all in, but I think I ought to ask you and Elio to classify at least a couple of conversational typos for the benefit of the other listeners. I'm sure that many people will have written to you about the assertion that the Cretaceous Carboniferous period was 400 million years ago, but some may not notice the conflation of water fern and duckweed. Elio did say correctly that Azolla was a fern, but this was after he already said it was duckweed. He clearly knows it isn't, but the listeners may not appreciate that.
Thank you for correcting
Good catch.
me. I appreciate being told.
Azolla is a serious invasive alien plant in UK wetland habitats and can quickly choke out almost everything else when it gets into ponds where there's nothing that can control it or compete with it. Even the duckweeds that might otherwise cover the whole surface of ponds can be deprived of the open water that they need and be replaced with an almost hummocky surface of piled up water fern. It can be quite a dismaying sight to go visit a pond from one's memory and then see as one approaches where it used to be the telltale red of the hummocky surface of water fern that has obliterated it. I don't know if anyone has yet come up with a safe and practical control method. I used to get a bit concerned when eutrophication had enabled the duckweeds to cover the whole water surface, but this Azolla is orders of magnitudes worse. These slips and typos are inevitable with such a wide-ranging and unscripted format. In a way, they are the deliberate "Where's Wally?" that encourage us to keep listening carefully. May they and you all long continue. Best wishes for the New Year. Kind regards, Steve.
Well, that's very generous.
It's lovely.
It's very generous. I certainly plead guilty.
And, this is really live radio. We don't go back and adjust and fix and tinker.
Only if we have to, if we really need to.
Yeah, we did it once when I couldn't figure something out and we had to cut it all out. Yeah.
Yeah.
But this is a really nice way of saying that our errors are actually educational or informative.
Well, sometimes it's conversational. I mean, we get into things we know nothing about.
Yeah.
Right? We pretend we do, but that's inevitable. Otherwise, the conversation would be very stilted. So we risk making mistakes, and we recognize that.
But that's the intent. We're having a discussion.
It's very interesting that many scientists are uncomfortable with this kind of format because they don't want to say things they're not sure of. And so it's not easy to recruit people to do these podcasts, and that's one of the reasons. In fact, one scientist told me she would like to give a talk from a script and go through it for their slides and never veer from it. So a conversation terrifies her, So it's and I can understand that a lot of scientists are like that. But I also love unscripted discussion. So
well, and I also love learning about what a great audience we have. Such a wide variety of people tune in. So I'm grateful.
Yeah, absolutely. Without them, we wouldn't be doing this, right? All right, that is TWiM 97. Ladies and gentlemen, we are creeping towards number 100.
I know. How will we celebrate?
I don't know. That's a good question. You can find all of them over at iTunes, also at microbeworld.org/TWiM. And if you would like to send us a question or comment, we love to get them. Send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. I'll see you next time.
You bet. Elio Schaechter is at the lovely blog Small Things Considered. Thank you, Elio.
Thank you. My goodness.
You must check it out. It's really cool. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
iChip, huh?
iChips.
Wow. Is there an app for that?
I'm certain that small biotech company in Massachusetts is looking to make one so they can plow through more samples of dirt.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.